Namaskar. Now I am going to discuss about mitotic cell cycle. What is actually the mitotic cell cycle? What are the different events, major events? What are the phenomena occur during the cell cycle, and what are the different parts of the cell cycle? We'll discuss about it. Now, if we draw a cell cycle, that is a cell, when it divides into 1 to 2, that is the mitotic cell cycle's main characteristic. When a cell is divided into 1 to its two cells with equal chromosome number, then before division during the mitotic phase, that is the M phase, it actually goes through a different phenomenon. Now, what is actually the cell cycles? Two different. If we divide a cell cycle into two major stages, two major part, what are the two major part of a cell cycle? It is a cell cycle, and it can be divided into two major parts. That is the interphase. Interphase. Here I have written interphase. Interphase. Interphase is actually, it is composed of three stages, substages, G1S and G2. This is the interphase. And another is M phase. M phase, that is mitotic phase, and this M phase is again subdivided into four parts: prophase, metaphase, anaphase, telophase. We know that a mitotic metaphase or mitotic phase, M phase, is composed of four parts: prophase, metaphase, anaphase, telophase. But what is actually the interphase? Interphase consists of three parts: G1S and G2. G1, that is a gap phase, gap 1. S is a synthesis phase. G2 is a gap 2 phase. Now we'll see that what is actually the phenomenon occur during the G1 phase. What is actually the gap phase? Gap phase is the phase where cell takes decision whether it will go to the next round of cell division or not. During this stage, this is the most vital stage, the largest stage, G1 phase, and this is the most vital step where cell decides, it's double its size. What is the event? What is the event of the G1 phase? G1 major events of the G1 phase, that cell organelle number. During this G1 phase, cell organelle number increases or doubled. Cell organelle, that is the different cell organelle, ribosome, lysosome, mitochondria, this is the number of cell organelles will be increased in number and produce raw material for DNA replication. What are the different raw material? As you know that the histone protein, polymerases, and the origin replication sites, that is the origin region, it is identified, origin region is specified by the origin pre-replicative complexes. As you know that the pre-replicative complexes are the complexes of which are responsible for the start of a replication, the origin site. So the proteins for the start at the origin site are synthesizing during the G1 phase. So G1 is a very dynamic phase at where the cell organelle number are increased or doubled, produce raw materials for DNA replication, that is the raw materials, the DNA polymerases, Pol enzymes, histone proteins, different other substances. As I mentioned that the pre-replicative substances like origin replicative complex, CDC6, CDTT, these are the different proteins which are necessary for the accumulation of or for the formation of pre-replicative complex. What is actually the pre-replicative complex? Pre-replicative complex is a complex which are accumulated at the origin site of DNA before the start of the DNA replication, but it remains in inactive stage till at the S phase, that is synthesis phase. So at the gap phase is actually the preparatory phase, whether the cell will pass to the next stage or not. If the cell accumulates all the substances for the synthesis, or which is sufficient for the synthesis of DNA, then the cell is allowed to go to the next stage, that is the S stage. So what are the major events here? First event is cell organelle number is doubled. Produce raw materials for DNA replication. As I mentioned, raw material, polymerases, different histones, different pre-replicative complexes and proteins are all synthesizing during the G1 phase. At the G1 phase, cell increases in size also. So these are the major events in the G1 phase. Now, from G1 cell, if the cell fulfills all the criteria of the G1 phase, then cell will pass to the next stage, that is the S stage, that is the synthesis stage. But in that stage, from between the G1 and S stage, there is a checkpoint. This checkpoint is called G1S checkpoint, and this is a very vital checkpoint. This is called start. That is, if one cell will pass this start point, then cell is committed to divide, and this is a very vital event. In case of cancers, many kind of cancers, if this checkpoint is mutated, the proteins present in this checkpoint, if get mutated, then the cell will run continuously and uninterruptedly. As a result, the formation of tumor. So this is the first checkpoint, G1S checkpoint. So what is actually the G1S checkpoint? We will discuss later. Now, in the S phase, what is actually the event in the S phase? S phase is the synthesis phase. As the name implies, it's a S for synthesis. Synthesis means it is responsible for the DNA synthesis. What? DNA synthesis, DNA replication. As I mentioned, that is the pre-replicative complexes are formed in the G1 phase at the origin site. In the S phase, replication starts, and what are the major events? The major events are initiation of DNA replication, CDK kinase phosphorylases, pre-replicative complexes. In the G1 phase, pre-replicative complexes are organized around the origin site, and in the S phase, these pre-replicative complexes becomes activated by different CDKs, cyclin dependent kinases, and get phosphorylates. Different CDK phosphorylates these pre-replicative complexes. As a result, this CDK kinase phosphorylates the pre-replicative complex and replication starts here. What is another phenomenon? That the different polymerases, polymerase delta in case of eukaryotes, polymerase delta, epsilon, and polymerase alpha primase. These different polymerases, polymerase delta, polymerase alpha, delta, polymerase alpha primase, and these different polymerase accumulates at the origin site and start replication during the S phase. So S phase is the phase where a single chromosome is divided longitudinally to form two chromatids with its main centromere, and DNA material becomes doubled in that case. So here, if DNA is 2C, here DNA will be 4C. DNA material is doubled. Now, the next stage is the at the start side of the S phase, at the start point, the centrosomes first divide. As you know that the centrosomes are responsible for the formation of spindle fibers, spindle tubules. So centrosome division occurs in the early stage of the S phase, at the early stage of S phase. So these are the different events what I mentioned here. I am repeating it. Initiation of DNA replication, different DNA polymerases accumulates, and pre-replicative complexes, different proteins present in the pre-replicative complex that becomes activated by different CDKs. Here the polymerase, and last stage here the it is that is the new I mentioned that is a duplication of spindle fiber forming substance, that is the centrosome occurred at this stage. That is the centrosome doubling of centrosome occurred at the S stage. Now in the G2 phase, what are the different phenomena of the G2 phase? As I mentioned, the G2 is a phase where it is the checkpoint, is the gap phase, where actually the DNA synthesis occurred properly or not. If DNA synthesis occurs properly, then only the cell will move from S phase to G2 phase and from G2 to M phase. G2 phase is actually a phase where proper checking occur, that either DNA breakage occur, DNA breakage is there, single strand breakage, double stranded breakage, what kind of breakage, or unreplicated DNA is present or not. This is the function of a G2 phase. However, what are the actual event of the G2 phase? In the G2 phase, G2M checkpoint, there is a checkpoint, as I mentioned, the first checkpoint and the second checkpoint is the G2M checkpoint. The G2M checkpoint checks whether DNA is replicated properly or not. By means of two proteins, ATM and ATR. ATM checks double strand breakage and ATR checks single strand breakage. Now, at the G2 phase, geminine protein, one protein is there, geminine. At the G2 phase, there is a protein geminine. That geminine protein inhibit another protein CDT1. This CDT1 protein is actually responsible for the initiation of replication, pre-replicative complex. If we carefully notice and going to the recap the G1 phase, then we will see that the pre-replicative complex, one important part is CDT1, that is initiation of replication occurred by the CDT1. It is a member of pre-replicative complex. So here geminine protein directly inhibits that the CDT1. Why? Because one replication event is running, and during this phase, during this stage, if another replication will start, then cell loses its control, and within a one cell, from one cell to two cell will develop with a double number of chromosomes. However, this is the phenomenon of G2. Actually, the major phenomenon of G2, we have little known about this stage. So after proper checking that the DNA is fine, DNA is replicated properly, no breakage is present in the DNA, either single strand breakage, double strand breakage, DNA properly replicated, then this checkpoint, G2M checkpoint, allow to pass to the next stage, that is the M stage, the most dynamic stage of the cell cycle. That is the mitotic phase or M phase, which has been divided into four substages: prophase, metaphase, anaphase, and telophase. Now, what is the event of the M phase? M phase, the major events are the so many events at the prophase, at the early prophase, chromosome condensation. First, the chromosomes start condensed at this M phase, first stage prophase. At the metaphase, at the prophase is the later stage of the prophase, that is the nuclear envelope broken. At the spindle fiber formation takes place from the centrosome, at the two end of the centrosome, nuclear. In case of animal cell, that is the spindle fiber formation starts. Is spindle fiber properly attached to the kinetochore or not? If spindle fiber properly attached to the kinetochore, whether the alignment of the chromosomes occur properly at the center region, whether they are aligned properly, or these are the major events, all checking occurred during this stage, that is the M phase. There is a checkpoint, metaphase anaphase transition checkpoint. It is actually the APC protein, APCC. This protein plays important role, and if all phenomena occur properly, that is the spindle formation occurs, spindle is attached properly to the kinetochore, that is the chromosomes kinetochore. I have mentioned in a previous video, that is the proper arrangement occurred at the metaphase. All the chromosomes arranged properly at the center region, all the spindle fiber attached to the kinetochore properly, and cell will ready to divide. Then only here metaphase anaphase checkpoint allow the cell to pass to the next stage. It is called metaphase to anaphase checkpoint. This is the most vital checkpoint. And APCC, anaphase promoting complex protein, plays an important role throughout this segment at this point. Now, so at the telophase, you know that is a very simple phenomenon. At the anaphase stage, chromosomes are separated. At the telophase stage, the chromosomes are going to the two different pole, to opposite pole, and the reorganization of the chromosomal, as you know, the reorganization of nucleus occur, nucleus formation takes place. So this is the most vital and dramatic event of the cell cycle. So already we have discussed about the different stages, different phenomena. What are the different phenomena occurred at different stages of the cell cycle? I have mentioned it properly. Now we will see that what is actually the function of the checkpoints, and what are the different CDKs, cyclin dependent kinases, and what are the different cyclins, which different cyclins are present in which stages of the cell cycle. Now here is a I have made a table where different kind of CDK throughout the cell cycle present. What are the different kind of CDK? I have mentioned here the G1 CDK. There is a G1, so G1 CDK is there. What are the different kind of G1 CDK? G1 CDK are CDK4 and CDK6. At the G1 stage, CDK4 and 6 is present. Along with these CDK4 and 6, D-cyclin is present. So CDK4 and 6 along with the D-cyclin, they together form a complex, CDK cyclin complex, which are responsible for the progression of cells to the next stage. So this is at the G1 CDK. What are the G1 CDK? G1 CDK is the CDK4, 6 and G1 cyclin is D-cyclin. So at the G1 stage, this CDK4 and D-cyclin is present for the proper functioning of the cell cycle. Now at the G1S checkpoint, which CDK is present? CDK2 and E cyclins together form a CDK cyclin complex. This CDK2 and CDK cyclin E, if properly work properly, then they will properly check the cell whether all the function occur in the G1 phase or not. If they are defective, then cell will pass without completing the phenomenon properly into the G1 cycle, into the S cycle. So cell will not fulfill, or cell will not properly divide. So this is a very vital cyclin CDK complex, that is the CDK2 cyclin E. So this is a checkpoint. At the S stage, there is a CDK1, 1 and A cyclin. At the S stage, CDK1 and cyclin A is present, and M CDK. At the M stage, there is another CDK there. M CDK is CDK1 again and B cyclin. This CDK1 and B cyclin together form a complex, so that cell whether the proper phenomenon occur, whether the phenomenon occurred during the M phase is occurred properly or not. So these are the different CDK: G1 CDK, G1S CDK, S CDK, and M CDK. So at the G1S CDK, at the G1S checkpoint, these two CDK complex, CDK cyclin complex, if not work properly or mutated at any stage, then the cell will run continuously and cell will form tumor. And another checkpoint is G2M. If these are mutated at the G2M checkpoint or M checkpoint, if mutated, then what will happen? That cell will run or cell will divide without proper alignment of the chromosome, or in the new cells chromosome number will be unequal. So this is the overall cell cycle here, with some amount of proteins at different cellular different stages and sub stages of the cell cycle. I have mentioned here the different phenomena of the cell cycle. What are the different events in the different stages of the cell cycle? Thank you.